So for all those undeveloped viruses, for the entry, now we call it classical virus fusion trigger. We have 4 modes. The first mode is a typical HIV. Typical HIV virus would bind to the receptor. The receptor could be CD4 or co-receptor XCR4 or CCR5. So usually this is type 1 single receptor ligand interaction. So it's a one protein in the HIV is a GP160. This protein will interact a receptor or a co-receptor. So they are single interaction for this step. We call it type 1. And the second one is a typical herpes virus. They need the coordination of multiple, the multiple receptor ligand interaction. Here again, all those two fusion between the cell membrane and the virus membrane occurred on the surface of the plasma membrane. So those are the 2 types, the first 2 types, herpes and HIV. Also SARS, MERS, also this type. And the third type would be flu. Flu is another example. There they have a interaction on the surface with a receptor, a specific receptor like receptor and the virus protein interaction. Here, after that, the virus will be endocytosed into the cell. And the flu virus is very clever. I call it a clever virus. The flavor and also flu virus is in a rush. They cannot wait. So they included their own protein called M2. So the M2 included by this flu virus will help the endosome to lower down the pH. To speed up the fusion, they need a pump to make sure this pH will lower down very quickly. So this is exactly what happened to flu. And then the virus will fuse with this endosome membrane. So fusion occurs in the endosome under low pH condition. This is type 3. And the type 4 would be dengue, Zika. It's another example. They also bind in the surface of cells, but without any, at least I call it, without known receptors. Maybe there's a receptor, but no one found a receptor. The young, virologist sitting here, molecular biologist here, cell biologist, go and find a receptor for any of these flavivirus so far, but there are some universal or non-specific receptor like TIM through the phosphatidyl serine binding or DC sign through the carbohydrates interaction. That's not specific. With this binding, the virus will get into the cell through the endocytosis. Again, here, dengue and all those flavivirus, the current theory or hypothesis, in comparison with the flu, it's a lazy virus. They are waiting for the endosomes to mature. Endosome will eventually mature into gradually becoming a lysosome. So the pH will be lowering down. So this, through the pH lower down here, again, will be endosome membrane and fuse with the virus membrane, and then fusion occurs. Genetic material was injected into your cell, and then the whole process will start for the replication. The virus. Once you get into the cell, that's the entry and the fusion. And then replication, the genetic materials. After that, they will assemble and release into a new virus particle. So those 3 steps are important. So my lab has interest for this entry and also a little bit for the exit. So far, all this, the structural basis is known. This is a good example for this structure. This is a type 1. It's a typical, it's a HIV, MERS and SARS coronavirus, and also for the HIV. So cut a long story short, it's like the protein first get bind to your receptor or cell surface like this way, hold on and touch the bilayer membrane. So after touch this, there's a conformational change for the protein. The protein encoded by the virus, and then with this conformational change, the protein will go like this, push down the cell membrane and touch the viral membrane like this. Really through this, this is the structure, and then through this will fold back here, put the 2 membrane together. So this is the whole process. And type 3 very similar. And the class 1 is the virus is the dengue Zika, yellow fever, all these viruses, it's very similar. On the surface of the mature particles, you have an unavailable protein on the surface. It's lying down like this way. It's lie down here. But once they get bind to the cell membrane, they also protrude out. And then, again, they have another conformational change to get two membranes together. This is structural basis so far with those three models. So now I will give you an example for what we have done and what kind of research we are doing and what kind of hypothesis we build up. And eventually, I hope it will be a theory, through the hypothesis into a theory. So the MERS is a good example. For the MERS virus, a typical undeveloped virus, you have an undeveloped, and you have a spike protein, protruding out on the virus surface, spike protein. And for this, they are also, looking for a receptor. There are a lot of molecules here. As far as we know, the MERS coronavirus as a receptor CD26, also DPP4, was defined by the Dutch group, Erasmus University there. And once they found this. Yeah, this is the, spike protein bind to a receptor. Yeah, this is the interaction. So my group, what we try to define the interface for this interaction. And first, how the CD26, the receptor, and they interact each other. And then we try to define the molecular basis, and that will lead you for the development of some inhibitors.